Hello, I'm Jezebel. I work in the School of Medicine in Swansea University. Here I do research in nanomedicine. Some of you may wonder what's nanomedicine. So, nanomedicine is the application of nanotechnology into medicine to improve the healthcare. Nanomedicine goes from the production of nanotherapeutics to treat and prevent diseases to the building of nanoelectronic biosensors to detect malignancies.
But what is nano?
Good question. Nano in Greek means dwarf. Nano size, it means that it's very, very small. We cannot see with our bare eyes something that measures nanometers. Even a traditional microscope cannot see a molecule that measures 1 nanometer. So, to help you understanding what it's 1 nanometer, you will travel with me along the different sizes. Him is 1 meter. Then, 10 times less is the width of my hand. 10 times less than this is the width of my finger. If I divide this by 10, is the size of the head of a needle. Then, if I get the head of a needle and I divide it by 10, it is the width of a diameter of a hair. Up to here is what we can see with our bare eyes. But if we get 1 hair and we divide the diameter by 10, we would reach the size of the red blood cells. Then, if we get 1 red blood cell and divide it by 10, we would reach the size of a bacteria, which is 1 micrometer. We are not yet in the scale of the nanometer. If we get 1 bacteria and we divide it by 10, we will reach the size of a virus, which measures around 100 nanometers. Then, if we get this teeny tiny virus and divide it by 100, we would reach the size of 1 nanometer. And 1 nanometer is the same as half of the width of a molecule of DNA. So then, 1 nanometer, imagine that is the size of this marble. Then, if this marble measures 1 nanometer, it means that 1 meter would be as big as the size of the planet Earth.
But why do we care about nano?
Good question as well. How the material behaves in the nanoscale is very, very different from how the things behave in the larger scale. Thanks to nanoscience, we know that the properties of the nanomaterials are very, very different, and we can use this knowledge to build better and more precise and more sensitive instruments, and also to generate better therapeutics. So, as the first experiment in these What's Nano videos, we will extract DNA and we will be able to see. Yeah, I know it's very small, but you will see that you will see. So, let's go. For the homemade DNA extraction, we will need the following materials: strawberries, a Ziploc bag, a coffee filter, dishwashing liquid, some little containers, ethanol or alcohol that you can find in the pharmacy, and something to start cutting and mashing up the strawberry. So, the first step will be to mash up a strawberry. And now, once the strawberry is cutted, I'll need a helper. Okay, we'll transfer the strawberry into the Ziploc bag. We'll close the Ziploc bag properly to not to have any spill accidents. And we'll proceed to the mashup.
Mashing completed.
Thank you. And now we will add about 5 ml of dishwashing liquid. Please help her. We will mix the dishwashing liquid with the strawberry mashed up. Let's do a good massage, a good mixing, so that the detergent can break all the cells that are mashed up here. Once we've given a good mix of the mashed strawberry with the dishwashing liquid, we will transfer the contents into the coffee filter to filter it and collect the rest liquid here. Once filtered, we will have sort of this messy liquid, and we will transfer a little amount into another container. We will get the ethanol or alcohol, and we'll add some drops. And we will leave some minutes for the magic to happen. After a few minutes, we will start seeing something like this, different phases, is it in the upper phase that we will find our DNA. Some more minutes and voila! Here it is, our DNA. So, you see that we've seen. Here we have our DNA, but why are we able to see this if it's such a teeny tiny molecule? Well, its diameter is very tiny, but it's a very, very long molecule because it needs to contain all the information for all the cells of our body, for example, or for all the cells in a strawberry. Then, to be able to fit inside a cell, it needs to be a really, really compacted molecule. It means that it needs to be self-assembled. That is what self-assembled means. Really organized to be really, really packed. And because here is condensed, we are able to see. So next experiment will be about self-assembly. See you soon. And now let's move to our second experiment. In this second experiment, we will see a little bit more what is self-assembly. For this, we will become molecular chefs and we will prepare some sweet gummy bits. For this, we will need the following ingredients. I love this magic. So, we will need sodium alginate, food coloring, sodium chloride, some room temp water, some warm water, blender, and other tools. Let's go. Sodium alginate. This comes from a brown seaweed that some of you may know that is called kelp. All over the world we can find this seaweed, and we can find as well this sodium alginate in many foods like ice cream, like sauces, syrup. It is a thickening agent. Then first we get 1.5 spoons and we'll blend. Once it's a homogeneous mixture, we will add the food coloring. We'll add 4 spoons and we'll blend it again. We'll leave the mixture sitting for 15 minutes for the bubbles to go out from the mixture. And now we'll use the sodium chloride. Sodium chloride is a salt, for example, similar to the one that you could use in your fancy baths. Then we'll add around 3/4 of a teaspoon into this bowl that contains 2.5 cups of water, warm water. So like this, the salt will dissolve very easily. After 13 minutes, the mixture should look something like this, without bubbles in the middle. We'll see what happens if we add the sodium alginate to water, or if we add the sodium alginate mixture to the sodium chloride bath. Then, if we get the sodium alginate with this green food coloring, and we add some drops to the water, we can see, well, nothing else than green drops on water is happening. But what if we add the sodium alginate with green food coloring into this sodium chloride mixture? Let's see. So, let's do a close-up to see that better. We can see that some spheres get formed when the sodium alginate get into contact with the sodium chloride. Well, we can even do big things like worms, gummy worms. So what is happening here is that the sodium alginate, when it gets into contact with the sodium chloride, it undergoes into a chemical reaction and self-assembles. Here is the word, self-assembles into these spheres or worms containing the green food coloring that we've added to the sodium alginate mixture. So we are indeed doing espherifications like molecular chefs. And if these components are food grade, you could even eat these little gummy bits. So now you know what you can add onto your next ice cream, sweet gummy bits. So now you've seen self-assembly in action and how cool it can be. Self-assembly happens in nature in many ways when molecules organizes to get some functionality. So we can see self-assembly in the snowflakes, in the soap bubbles, or even in the DNA, as we've seen in the first experiment. But why we scientists cares about self-assembly apart from these cool gummy bits? Okay, so in nanomedicine, we use sort of a very similar process to build nanoparticles, so exactly the same as this, but in a nanometer size. And instead of getting food coloring inside these espherifications, inside these nanoparticles, we get some drugs to treat, for example, cancer. And if we sort of program these teeny tiny nanoparticles to go where we want them to go, voila, we have the perfect medicine. That's why we care about self-assembly. And well, I hope you've liked these experiments. See you soon.